Good day, everybody. Thank you for attending this webinar. In this first module, I'm going to be focusing on a little bit of background on SciCon innovation, as well as the technology behind the SciFlow 1000 fluidic culture system, pictured here in the center of the slide. For additional information, if you want to contact us, please visit our website or email or telephone us directly. Thanks. I'm going to get started. A little bit about SciCon innovation, what we do. There are three parts of our business. There are three facets to our business. Historically, since about 2009, we've had a skin and tissue procurement business, and we also have a proprietary line of media that we sell called cryothaw, which is very good at freezing down cells, leaving the cells in a viable state so that when they're thawed out, they recover and function in a biologically relevant fashion. Additionally, we have CRO services. So we run projects for customers and we deliver data or results back to those customers. Predominantly, though not exclusively, the CRO services are focused at demonstrating the benefits and features of the SciFlow 1000 fluidic culture system. We also have customs tools business where we help people design novel fluidic systems as well. For the rest of the talk, I'm going to focus in on the SciFlow 1000 as that is the core business moving forward for SciCon innovation. What are some of the advantages of the SciFlow 1000? So there's three areas I'm going to focus in on. The first being biological relevance. I'm going to get into more information about SciFlow as the talk goes on, but SciFlow 1000 is a fluidic culture system that connects together multiple wells in series in a standard tissue culture plate. With those multiple connected wells, one of the things you're able to do is you can connect cells of different types together in series across the plate. So you could have upstream liver cells, you could have downstream target organ of interest, you could have downstream off-target cells as well for looking at a whole biological metabolism, toxicity, efficacy type of study. Additionally, because it is a fluidic system, we have gradients. Gradients are a key feature of many biological systems. We get gradients of compounds, gradients of metabolites, gradients of cellular responses. I'm going to go into a lot more detail on gradients in the applications portion of this talk. Compatibility. Because the SciFlow 1000 is an SBS compliant 96 well plate, we are compatible with standard plate readers, standard assays, standard high content imagers that are available on the market. So while it's a new system, there's no need to change your workflow. There's no need to change the technologies. There's no need to change the instrumentation and kits that you're already running in your lab. And last but not least, the system is very data rich. What I mean by data rich is you can generate a lot of information very quickly and efficiently from a few number of SciFlow 1000s. We do a lot of high content imaging. We do a lot of real-time analyses. So all the data shown here in this last panel comes from a few rows of one SciFlow 1000. So each plate tells a story. There's a lot of information you get from each of your experiments. So to summarize, the benefits are a biologically relevant system, usability and compatibility, and a data-centric system. To take a small step back from the technology, at the heart of the SciFlow 1000, it is a surface, it is a system for culturing cells. I'm now just going to show a small set of cells that we've cultured on the SciFlow 1000. On this slide, I'm highlighting it on human cells. SciCon has a long history with liver cells. Our founder has a long history with working with liver from multiple different species. What I'm showing on this slide are human hepatocytes at day 0, human hepatocytes on day 1, some non-parenchymal human cells, as well as some work we've done with breast cancer or MCF7 cells. There's many other human and non-human cell types we've also successfully cultured on SciFlow. I also want to mention that SciFlow 1000 is compatible with collagen coatings, it's compatible with Matrigel, it's compatible with fibronectin. There's a lot of different surfaces that you can put on SciFlow for culturing specific or alternative cell types. A lot of the work I'm going to talk about in the applications portion of this talk focuses on HEPA-RG cells. HEPA-RGs are a more biologically relevant cell line for looking at liver metabolism and liver safety and efficacy studies. What I'm showing here is a 24-day time course of culturing the HEPA-RG cells in the SciFlow 1000. I also want to mention all the images I'm showing are directly in the SciFlow 1000 and visualized with a standard inversion microscope. So there is no special instrumentation you need for looking at your cells, for checking on your cells, for grabbing quick pictures of your cells to make sure everything is progressing normally and the way you want it to move forward. I mentioned SciFlow is also compatible with high content imaging. I wanted to put in this image of a little bit of fluorescence imaging work that we did. These are some HepG2 cells that I'm showing that are stained with Herc stain, which is shown in blue, and Celltox green, which is shown in the pinkish reddish color, and it's just measuring cell viability directly in the SciFlow 1000 in a Molecular Devices Image Express high content imaging system. Because it is a fluidic system, we often get asked, are the cells healthier? Are the cells more metabolically competent? Do the cells look more biologically relevant? What I'm showing here on the top two panels are day 1 and day 14 images of the HEPA-RGs in the SciFlow system. On the bottom, I'm showing again day 1 and day 14 of those same HEPA-RG cells cultured in static plates. Visually, they look very similar. The cells in SciFlow, the cells in static, they have a similar morphology, similar cell phenotype as we're looking with our standard inversion microscope. We also measured cell titer glow, or ATP, on day 14 in both the static and the SciFlow systems, and we can see relatively similar amounts of ATP in both the SciFlow and static systems on day 14. We also looked at GSH. GSH is a measure of reduction potential. We did this with the Promega kit GSH Glow, and we can see that there's a roughly 50% higher level of GSH in our SciFlow, in our cells cultured in our SciFlow plates than the cells cultured in static plate. So the cells do appear healthier, they do appear a touch more metabolically competent, or at least phase 2 metabolically more competent in the SciFlow versus the static system. I now want to focus on the SciFlow technology. How does the system work? And then I'll get into some of the applications or benefits of the SciFlow system in the second module. When people first get a SciFlow 1000 in their hands, they sometimes tell us that it looks complex. It looks like there's a lot going on. Well, one of the things that SciCon has done is we've engineered simplicity into the system. There is a lot of CAD work behind the scenes. There's a lot of fluid mechanics. There's a lot of material science that went into developing the SciFlow 1000, but the end result is a very user-friendly solution which is ready to use right out of the box. It comes pre-packaged, individually sterilized, tissue culture treated, and ready for you to culture cells directly in the SciFlow, directly in the system. SciCon has handled the engineering so our customers can focus on the biology. I also want to mention, not pictured here, the SciFlow 1000 does come with a lid. It comes with a lid to maintain sterility and allow you to move the plate around the lab without opening it up and exposing it. I just didn't have the lid in this picture because it made it more challenging to see the system. I've mentioned multiple times that the SciFlow 1000 is a fluidic culture system. So the question is, what drives the flow? What causes the flow from well to well across the plate? What I'm showing in this image is a side-on view of a SciFlow 1000 with a blue indicator dye in one row of the plate. What I'm highlighting here is the 0.5 millimeter or 500 micron decrease in well height as you move from left to right across the plate. It's this decrease in well height that drives the fluid flow. It's this hydrodynamic pressure that causes the SciFlow 1000 to flow from left to right across the plate. All of this flow is accomplished with no pumps or no tubing. The SciFlow 1000 is made entirely out of one material. It's made entirely out of tissue culture treated polystyrene. And as I said before, but it's worth repeating, it is an SBS compliant 96 well footprint. So it works with your standard plate readers, high content imagers, multi-channel pipette man, and laboratory robotics that you may already have in your lab. Gradients. I mentioned gradients on an early slide. This is a top-down view of one row of a SciFlow 1000. Not shown all the way on the right side of the plate is a well or a sink or an overflow well in column 12. But I want to highlight column number 1 or well number 1, which is the dosing well or the source well. This is a larger well. It accommodates approximately 500 microliters of media, and it allows for uninterrupted culture for longer periods of time. So what you see across your SciFlow 1000 is you get a source well on the left, a sink on the right, and then 10 identical culture wells in the middle of the plate. There's 10 identical wells for culturing cells across the SciFlow 1000. Each of these wells is the same size as a half area 96 well plate. When you're using the system, you're adding compound or drug into the source well, and the fluidics causes that to flow across the plate from left to right. What this does is this generates a gradient. You get a gradient of test compound. As the test compound flows across the plate, it dilutes into each well of the plate and then continues flowing downstream or downhill. So you get a decreasing concentration of your test compound from left to right across the row. Additionally, if your cells are able to break down that test compound, you get metabolites generated. Those metabolites also flow across the plate, but what you get is you get an inverted gradient of metabolites. As the metabolites are being generated uphill or upstream, but continue to flow across the plate, you get accumulation of those metabolites in the downstream wells. So essentially what we get is we get a separation. We get higher concentrations of test compound upstream and higher concentrations of metabolites downstream. So where we see a certain effect, in what wells of the plate we see a certain effect, give us information about what is causing that effect. If we see cell death, if we see efficacy of our compound in the upstream wells, we know it's a compound-mediated effect. If we see our effects in the downstream wells, we know it's more of a metabolite-mediated effect. So there's a lot of mechanistic information we get from SciFlow in addition to just the compound itself and the safety and efficacy of that compound. Exposure and fluidic performance. We frequently get asked, how does SciFlow work? What do the exposures look like? How does the compound flow across the plate? So what I'm showing here are two different schools of thought, two different ways of dosing cells with compounds or drugs. On the right should be very familiar. It's a static system. When you add a drug or compound, that drug or compound immediately goes from 0 to 100% and then sits at that same concentration for the entire study. That's ignoring cellular metabolism, but cellular metabolism is usually a small percentage of the overall drug concentration that you're looking at. If you want to investigate different drug concentrations, you can manually set up your own dilutions, and you can do 2-fold or 3-fold dilutions across the plate to look at various drug concentrations, each within its own well. It's a fixed concentration, and it's a known concentration. Contrasting that with SciFlow and its fluidic exposures, what I'm showing here is a time course across multiple days in multiple wells within a SciFlow that are all connected. What you see with the blue line, or a very upstream well, is there's a rapid increase in compound concentration across a few days, and then it reaches an equilibrium where it sits for the rest of the study. In well 7, or somewhere in the middle of the plate, it's a much more linear exposure that we see, as it takes time for the compound to flow across the plate from the source well until it does finally accumulate way downstream, or days 7 or 8 later in the experiment. Last but not least is a well all the way at the right side, or the downhill end of the plate. There's a longer lag phase and then a slower increase in drug or compound concentration in those downstream wells. So what we're seeing are very different, very unique dynamic concentration gradients over time. I do want to stress that the concentration gradient is tunable by the end user. How much volume you add into the source well and how frequently you add more compound into the source well affect these gradients, affect the way the flow goes, and affect the exposure of the compound. So you can expose cells to higher or lower concentrations as may be dependent based on the particular experiment. We frequently get asked, how fast does it flow? How far does the compound flow? How many doses do I have to give so my cells see my compound of interest? Shown on the top panel is a picture of a SciFlow 1000, again top down, and we're adding a blue dye into the source well just to visually show you how the dye is flowing across the plate, diluting across the plate over time as it flows from the source well downhill into the sink or waste well all the way on the right side. In the lower panel in the graph, I'm showing a fluorescein titration experiment, or a fluorescein flow experiment. It's a 60-minute flow, and what I'm showing is upstream wells and then more downstream wells, so you can get a sense of how the flow goes across the plate. What we generally see, or what we always see, is there's a quick burst phase where it takes about 15 minutes of rapid flow, where there's a rapid increase in drug or compound concentration in the upstream wells, and that 15 minutes is followed by a much slower flow, which persists for about 90 minutes to 2 hours, where there's a more gradual increase as the compound is slowly spreading across the plate. Beyond about 2 hours, there is a slow diffusion-limited process that's driven across the plate based by evaporation and some of the, there's a wick in the waste well. The wick also helps to draw a little bit of material across the plate, but it's a much slower diffusion-limited process. So the flow itself lasts for about two-ish hours in a somewhat rapid format, and then it's much slower and will sit there until a user intervenes, until a user adds more compound, adds more media into the source well to keep the flow going. I do want to stress, though, that the equilibrium volumes in all of the wells always keeps media in all of the wells, always keeps the cells covered with media. It does not dry out, so you do not need to intervene with the system every few hours. You can easily go once a day, twice a day, even 3 times, once a day, every other day, every third day, if required by the experiment. Applications. There's 3 different applications I'm going to talk about in this webinar. I'm going to talk about 2 liver injury or 2 DILI, one focused on aflatoxin with direct compound effects, one's focused on acetaminophen or APAP with metabolite effects, and one focused on bioactivation of a panel of chemotherapeutic agents, which involves stringing together upstream intestinal cells and, upstream liver cells and downstream MCF7 or breast cancer cells. Aflatoxin. This is a 5-day study that I'm going to talk about, and the readout is cell tox green or viability. It is a content imaging study. This study works with HEPA-RG cells, and the images that you see down the middle of the plate are the images that we generated directly out of the SciFlow 1000, directly using the Molecular Devices Image Express system. What I'm showing here in this data panel is a time course. It's about a 4-day study. Across the horizontal, you'll see the different wells within one row of a SciFlow plate. And what we did is we took our cells, we added in Hirsch stain and cell tox green. So you can see the blues, all cells, those are the Hirsch stain dyes. The pink or red cells are the cell tox green, those are the dead cells. And what we're looking at in our table is cell viability, or the percentage of cells that are alive. You can see at time 0, there's very good viability across the entire row. Because of the biological gradients that are generated in SciFlow, we see a gradient of our aflatoxin. The upstream wells see a higher concentration of aflatoxin. The downstream wells see a much lower concentration of aflatoxin for most of the study. So what we can see is that in the upstream wells, we see a precipitous decrease in cell viability based on exposure to the aflatoxin. In the downstream wells, we see much higher percentages of viable cells for a longer period of time because they're being exposed to much lower concentrations of aflatoxin, much lower concentrations of the taxane. So what we're seeing here are readouts or the effects of the gradients that we see within SciFlow. And we understand that the toxicity coming from the aflatoxin is a direct effect of the parent compound. We know that because the effect that we're seeing, the cell death that we're measuring, tracks with compound concentration. Higher concentrations of aflatoxin upstream, higher percentages of cell death, lower percentages of viable cells. Higher percentages of viable cells downstream, lower concentrations of aflatoxin. Not shown here, there was also a control plate that was just treated with vehicle or DMSO. None of these gradients are detectable in that study. There's the same similar high percentage of viability over the whole time course of the study. I also want to stress that this is a real-time experiment, so we had Hirsch stain and cell tox green present in all wells at all times. So what we're doing is we're taking the plate, putting it in the high content imager, visualizing it, then putting it back in the incubator for more time, reading it again, and just reading it at multiple time points over the course of the study. So the gradients are developing, the data is developing, the data all comes from a few rows of one SciFlow 1000 system. Turning away from the aflatoxin study to an acetaminophen study. This is a 7 and 9 day study. It also occurs with the HEPA-RG cells that were used in the last experiment. The readouts in this case are cell titer glow, as well as there's some microscopy data behind the scenes, but I'm not going to be showing the microscopy data in this presentation. It's also a comparison between SciFlow and static dosing. In this data slide, what I'm showing is a bar graph of SciFlow as well as static cells normalized to percent of DMSO control for cell titer glow or ATP levels. Across the horizontal axis, I'm showing well number. In SciFlow, this is distance from the source well. In static, it corresponds to a twofold dilution series that we manually generated from 5 millimolar of acetaminophen down to no acetaminophen. And what we're showing here in our SciFlow in blue, in our static in red, is that in our static cells at day 7, normalized to DMSO control, we see maybe a 20% decrease in cell viability at the two highest concentrations of acetaminophen, but that effect mostly dilutes out and then the cells look just like the DMSO or vehicle control cells for the rest of the dilutions that we created in our static plate. To contrast that with our SciFlow data, shown in blue, we do see a similar-ish 20% decrease at our highest acetaminophen concentration, but then very interestingly, we see a decreased viability or higher numbers of cell death downstream of that highest concentration of acetaminophen. So we're seeing a decrease in viability at lower acetaminophen concentrations, but then the cells do recover all the way down at the end of the plate. What we're tracking here is we're tracking the flow of metabolites. These effects that we're seeing in wells 4, 5, 6, 7 are not mediated exclusively by the acetaminophen, but by the metabolites of acetaminophen, which are being generated in the upstream wells and then flowing into the downstream wells and mediating their effects. I mentioned this was a 7 and a 9 day study. Look at the data on day 9. Again, to go back to the static for 1 minute. At day 9, we now do see a very potent effect with acetaminophen based on a 5 millimolar dose. We see almost no viable cells left. And then it dilutes out across the plate, where we're seeing not much of an effect at all in the way downstream end of the plate. To contrast that with SciFlow, we're again seeing a modest effect at the highest concentrations of acetaminophen, but then we see a much more pronounced decrease in ATP, a much more pronounced decrease in cell viability in the downstream wells where the metabolites of acetaminophen are flowing, accumulating, and mediating their effects. So interestingly enough, we see very different results between SciFlow and static culture conditions. And in the SciFlow condition, we're able to get mechanistic information. We're able to learn that the effects we see are not at the highest parent compound concentrations, but at the higher concentrations of metabolites, or the higher concentrations of the cellular responses that are being generated and flowing downstream. We also did some LC-MS work. So we did quantify both parent compound as well as the primary metabolites associated with acetaminophen metabolism in the liver cells. And what you can see is it validates the data, it validates the story I was telling. We see our highest concentration of acetaminophen or parent compound in the upstream wells, and then that concentration decreases based on dilution and flow across the plate. We see an opposite effect with the metabolites. We see lower concentrations of metabolites in the upstream wells in the SciFlow plate, and then the metabolite concentrations accumulate, they get higher in the downstream end of the plates, and those metabolites are at their highest concentrations in the wells where we're seeing the largest effect, where we're seeing the lowest percentage of cell viability, the highest percentages of cell death. LC-MS is a very useful technique for calculating or measuring both parent compound as well as metabolite concentrations in the SciFlow system because both concentrations are changing dynamically over time. A summary of the acetaminophen study. As I mentioned, it was a 9-day study, and what we saw APAP toxicity was mediated by a toxic metabolite, and the gradients generated in SciFlow, the separation of parent compound and metabolites in SciFlow, allowed us to understand what was causing the toxicity. That information was masked in the static plate because there is no flow. We do not know what is causing the effect. We don't know if it's a parent compound or if it's a metabolite effect that's mediating that effect. It also took a longer time to see those results in the static plates because when the cells were fed every 48 to 72 hours, we were effectively removing the metabolites, so the metabolites did not have as much time to accumulate in those wells and mediate those effects. CYP substrates, we often get asked, are the cells metabolically active? Do the liver cells still show native CYP metabolism? This is a 3-day study, and what we used in this study is we used some fluorescent CYP substrates. There are some CYP substrates that become fluorescent once they are metabolized. So they're not fluorescent at the start, but once they get broken down by the CYP enzymes, they become fluorescent. And this study is working with primary human hepatocytes as opposed to the HEPA-RG cell line. What I'm showing here is this is a 72-hour study focused on CYP3A4. And what you're looking across is you're looking across a SciFlow plate, so columns 3 to column 11. What you see early on is there's relatively low levels of those fluorescent CYP substrates, but as the cells are cultured for a longer period of time, the metabolites are accumulating and the metabolites are starting to flow downstream. So you're seeing lower levels of metabolites in the upstream wells, and you're seeing an accumulation of those metabolites as they're flowing across the plate. I do want to reiterate this is only a 72-hour exposure. It's a relatively short study. If the study were continued for a longer period of time, these metabolites would continue to be generated, continue to flow, and accumulate even farther downstream the plate. The APAP study that I showed was a 7 and a 9-day study, which is why the effects were much more pronounced in the farther downstream cells, downstream wells, as opposed to the upstream wells. One more example of CYP metabolism data. This is looking at a whole family of different CYP fluorescent substrates, again in our primary human hepatocytes. The primary human hepatocytes had higher levels of certain CYP metabolizing enzymes, lower levels of other CYP metabolizing enzymes, but again you can see that the metabolites are being generated upstream and then flowing into those downstream wells, and we see that same type of effect of flow and of metabolites accumulating downstream farther away from the location where they're generated. The last data set I wanted to talk about, it's that chemotherapeutic study that I wanted to talk about. It's again working with HEPA-RG cells. This is it's a 2-day study, and our readout is cell titer glow. To set up the study, what we're looking at is there's 3 different plates that we ran in this experiment. There's an experimental and 2 control plates. The experimental plate has upstream liver or HEPA-RG cells, followed by downstream MCF7 or human breast cancer cells. Control plate number 2 is just HEPA-RG cells. Control plate number 3 is just MCF7 or breast cancer cells. Shown at the table at the bottom is the panel of all the drugs as well as DMSO control, which we used for treating these cells. Circled in red is this one column. This one column is where all of the data on the subsequent slide was measured. All these cell titer glow data or ATP data was measured in this one column of all 3 SciFlow plates. So what are we looking at? So what we're showing here is our panel of drugs. In blue is our control only liver cells. It's measuring toxicity of the panel of chemotherapeutics on liver cells. In red is only MCF7 or breast cancer cells. So it's showing the toxicity of the panel of chemotherapeutic agents on breast cancer cells in the absence of any metabolism, in the absence of any bioactivation. Shown in green is the combination or organ on a chip situation where we had upstream liver followed by downstream MCF7 or breast cancer cells. So what you can see, by and large, in most of the drugs, the liver as well as the breast cancer cells, the blue and red lines, are showing values very similar to 100%. This is normalized to DMSO control. So they're showing very little decreases in viability when compared to DMSO treated cells on their own. To contrast this, when we look at the combination of liver upstream of breast cancer, in 5 of the 7 cases, we see a greater than 50% decrease in cell viability. We see a large bioactivation, a large increase in efficacy of 5 of the 7 different chemotherapeutic agents that we tested when those agents are first passage through liver cells before being exposed to the breast cancer cells. The liver cells are metabolically active. The liver cells are able to break down the chemotherapeutic agent that we added in there, and that chemotherapeutic agent is able to mediate its effect in a much more effective manner once it has been bioactivated by the upstream liver cells. A quick summary. I'm not showing all the data that's been generated on the SciFlow 1000. I focused in on some cell titer glow data as well as some high content imaging. There are many other assay kits that we've also run and tested on SciFlow. There's other dyes that we've used when looking at SciFlow high content imaging study. We've tested many different plate readers. We get customers who ask us, is it compatible with my plate reader? Is it compatible with this plate reader? So we've tested a family of different plate readers from TCAN, Biotech, BMG, as well as Molecular Devices. A lot of the high content imaging data shown in this presentation comes from the Molecular Devices Image Express system. We also have experience with the Thermo Fisher Solomics imager, as well as the Biotech, which is a combination plate reader high content imager. So we have tested many different plate readers, high content imagers, as well as dyes and assay kits. To wrap up, some of the benefits that our customers tell us they're seeing from the SciFlow 1000 is because it is a fluidic system. They see improved cell health and viability on many of the different cell types that they tested in the culture system. They love the biological relevance, and that's both the ability to have multi-organ systems where they have different cell types talking to each other across the row, as well as the gradient concentrations of compounds, drugs, cellular responses, as well as growth factors for looking at cellular differentiation. And last but not least, the broad compatibility of the SciFlow 1000 and the fact that it interfaces with the technologies, the instruments that they already have up and running in their lab really makes it makes for a very easy to use, user-friendly system that can be adapted very quickly to an individual laboratory environment. Again, if you have additional questions or want to reach out to us, you can look at our website, or you can email us at info@SciCon Innovation, or give us a call. Thank you very much.